Hello, my name is Pallavi Iyer. I'm a pediatric endocrinologist at the Department of Pediatrics, Medical College of Wisconsin in Milwaukee. Today, I'm presenting our collaborative work on investigating the oncogenic variation of pediatric differentiated thyroid cancer from FFPE samples using RNA sequencing and whole exome sequencing. As you know, thyroid cancer accounts for 11% of cancers in adolescents and 2% in children. And the most likely pathology is papillary thyroid carcinoma and follicular thyroid carcinoma. And they usually present as more aggressive in this population than in adults. In our study, we set out to identify genomic characteristics of DTC using both bulk RNA-seq and whole exome sequencing of formalin-fixed paraffin-embedded thyroid tissue from patients diagnosed with either of these two cancers from UAB and Children's of Alabama collected between 2017 to 2020. Further, we perform integrative analysis by using C-by-portal as well as evaluating patients' clinical symptoms and phenotypes and analyze each case for genomic variants of every tumor normal pair. In Table 1, we present overview of the characteristics of demographic, clinical, and ultrasound characteristics for our patients with papillary thyroid carcinoma and follicular thyroid carcinoma. Further, we talk about surgical pathology and TNM scoring for cases with PTC. Going on to our results, I will be concentrating on Figure 2 and 5. In Figure 2, we did differential expression analysis from RNA-seq data, which is a statistical method that identifies genes that are expressed differentially between two or more groups of samples. Here is a heat map showing expression of selected genes of interest for tumor PTC versus tumor FTC samples. The genes are colored according to their expression levels, with gold indicating upregulation and blue indicating downregulation. Loss of expression of BRAF was seen in the majority of PTC samples as expected based on DNA findings. Reduced expression of KRAS, PTEN, and NRAS was seen in PTC as compared to FTC samples. Reduced expression of thyroid-specific genes such as TSHR was observed in many samples. I will go on to Figure 5. We also did whole exome sequencing data. After performing stringent quality control, we conducted variant analysis. Here's an oncoprint showing the distribution of variation among the 6 subtypes across a subset of our genes of interest, shown on the right side. We identified 4 types of mutation: germline cancer, thyroid-specific, somatic, and homodeletion copy number variant. Specifically, ATM, BRAF, MAP3K1, MET, MSH2, mTOR, PIK3CA, and SDHA variants were identified in PTC cases, while BRCA1, IDH1, and PTEN variants were seen in FTC. DICER1 variants were seen in minimally invasive FTC and follicular adenoma. HRAS and PMS2 variants were seen in NIFT-P. No pathogenic variants were noted in KRAS, TP53, or TERT in our population. Pathogenic variants in DUOX2, TSHR, and thyroglobulin were also identified in some cases. We are also working on RNA-seq-based fusion analysis and have shown in Figure 4 a particular FTC patient case. In conclusion, this is the first study describing genome-wide genomic and transcriptomic analysis in a well-phenotyped cohort of pediatric thyroid cancer patients from the southeast region of the United States. We have identified notable variation between tumor and normal samples as between histological subtypes. In some cases, findings recap known knowledge, but novel associations were also seen. These preliminary findings expand our understanding of molecular mechanisms underlying tumor development, progression, and response to treatment. Follow-up on known or novel associations may aid in development of individualized treatment plans for this patient population. Analysis of variation identified in our RNA-seq and whole exome sequencing dataset is ongoing, with focus on analysis and interpretation of RNA-DNA correlations, statistical comparisons across tumor subtypes and pathologic findings with comparison to external data sets, and pathway-based analysis to study the likely molecular impact of the variation seen and to study higher-level network-related perturbations. I would like to acknowledge that this work was supported by the Call Pediatric Research Institute grant from the Children's of Alabama and Dr. Worthy's Lab Startup Funds. Thank you.